So our next speaker, which is currently located, as far as I know, in Switzerland, but she's an MIT professor in electrical engineering and computer science department, like Bonnie Berger and myself. This is Professor Caroline Uhler. Caroline, it's all yours.
Thank you very much, Regina, for having me here. So I'm very excited to tell you about some of our recent work. The first paper that I'll talk about is about the biological hypothesis of what links SARS-CoV-2 and aging together, which is based on work with my longtime experimental collaborator, G.V. Shivashankar, who is a faculty at ETH Zurich. And that then led to this hypothesis actually bringing it forward in a computational way to actually look at drug repurposing for SARS-CoV-2, then led to this joint paper with my amazing students and together with my experimental collaborator, Shiva. Okay, so let me start by telling you a bit more about the biological hypothesis that really guided us in the selection of data sets, etc., afterwards for the computational work. So I think we all know by now that SARS-CoV-2 pathogenicity and morbidity is very strongly age dependent with much more severe outcomes in the aging population. And the main or the prevailing hypothesis for this is the aging immune system, as I'm sure many of you have read and are probably also doing research on this. Now in this recent Nature Reviews paper, we described or we proposed an alternative or an additional way of how this actually could come about, which is based on the following well-known fact that what happens in our lungs is that they become stiffer, the tissue becomes stiffer with aging. And this is primarily due to the cells in the connective tissue that they show increased matrix deposition. And now, why is this all important? Well, we have these epithelial cells, which are some of the first cells that come in contact with the virus. And these epithelial cells, they actually change their state. When the tissue underlying it becomes stiffer, they go from an epithelial state to a more mesenchymal state. Now, again, why do I tell you all of this? Well, in Shiva's lab, in experiments, they showed that, well, if you have these cells on a more relaxed state, so they're more in the epithelial state, or if you have them on a more stiff substrate, so they're more in the mesenchymal state, so this is more like in the aging population, this is more like in the young population, these cells actually turn on very different downstream genes or gene expression programs when they are confronted with a cytokine, which is the similar signal as the viruses send out. And so the hypothesis that we have is that, well, maybe, and what we know, of course, is that the virus makes use of human cells, right, for their replication and human programs for their replication. And so the hypothesis that maybe, the virus sends out the same signal in aging and young cells. However, the downstream programs that are turned on are very different. And this could actually lead to this, very different outcome in terms of also replication of the virus inside our cells. And just to get a first, what is this hypothesis based on? Well, what we looked at is at these genes that are differentially expressed in the two different states, like whether it is on stiff substrates or on relaxed substrates. And you actually see that many of these genes that are differentially expressed here are actually, in fact, also the ones that are differentially expressed by SARS-CoV-2. Okay, so this really suggests that, if you want to find drugs for the repurposing against SARS-CoV-2, well, what is ideal or what might actually, nicely cut down the number of drugs that you're looking at is if you look at drugs that intersect or that target proteins that intersect the SARS-CoV-2 pathway as well as the aging pathway. And so this is really what motivated us to, choose the data sets we chose for the downstream analysis in terms of drug repurposing. So let me get to that. So, now, of course, based on this hypothesis, what we do is we take, and this is the amazing thing that Regina mentioned, we have to build on the data that just comes out, right? So here we were lucky that gene expression data just came out of SARS-CoV-2 infection in a couple of different cell types. So in particular in these lung cancer cell lines, these A549 cells, which are the ones that we're using here. And so what you can then find or identify are the genes that are differentially expressed with SARS-CoV-2. And you can do the same to find differentially expressed genes with aging. Okay, so there are large data sets for that, right? And for example, in the GTEx database. So now you can intersect these to actually find genes that are in the intersection of the two pathways. And so these are these genes here that we then looked at a little bit more in detail. And you see, and this is quite remarkable, right, that you see that in these genes at the intersection, they are either both upregulated with SARS-CoV-2 or aging, or both downregulated. And there are very few that show, a different kind of regulation. Again, hinting at the fact that there's really strong interactions between SARS-CoV-2 pathways and aging pathways. Okay, so this is all just at the gene level, right? So now we really want to identify the signature of SARS-CoV-2 infection and aging, and not just, which genes are differentially expressed, but actually what is the signature, what is the effect of this infection. And again, looking at the same data sets, they actually have multiple different data sets in there, different kinds of batches, different kinds of amount of infections, so different MOIs of the SARS-CoV-2 virus. And so looking at this, you can identify the direction or the signature of the disease. So this would be, here we're showing it in a particular embedding, but you can identify the signature of the disease that goes from normal cells to infected cells for different MOI values, or with or without the addition of this ACE2 supplement, which we all know is quite critical for SARS-CoV-2 infection. And you see here that what is nice is actually that this is quite robust among all these different data sets, that actually the signature is basically always pointing in the same direction. So this is how we identify the signature of SARS-CoV-2. Now, in terms of drug repurposing, well, a standard approach to what you want to do in order to prioritize drugs is you want to find drugs that point the other direction, right? So that you can revert the effect of the disease. And this is a pretty standard way of doing it, and I'll show you how we innovate this on the next slide. But basically, maybe first telling you about the data sets that we're using. So here is this is an amazing resource from the Broad Institute, which is a CMAP data set, which has millions of samples and thousands of different perturbations. In particular, they have 800 FDA-approved drugs, which are the ones that we used. And these were applied to many different cell types. So here the colors actually represent different cell types. And in particular, one of the cell types that is studied in this data set is this A549 cell line. So that's one of these cell types. And so now in the simplest analysis, what one would do is just, hey, we look at the effect of each drug in this cell type, we look at the effect of the disease, and we want them to be anti-correlated, right, as much as possible. So that's what we're showing here. So you want a drug that points kind of in the reverse direction of the disease signature. Okay, now, of course, in the body, we don't care about A549 cells, right? These are cancer cell lines. We care about the effect of these drugs on infected, normal epithelial cells, right? And these are not in the data set. So what you really need is somehow to figure out the signature of a drug that generalizes to different kinds of cell types, right? And in particular, the cell types that we care about. Also, if we just would use A549 cells, we're throwing away all this other data, right? And not all drugs have been applied to A549 cell lines. So you really want something where you can use all of the data together to actually look at as many of these FDA-approved drugs as possible. Okay, so how do we do this? How do we actually figure out the effect of a drug in a way that is generalizable across many different cell types? So if you think about this problem just from a machine learning point of view, this really kind of smells like a style transfer problem, right? So what we have here, we have different cell types, and we have cell types where we know the effect of a drug, which would be here. And we have another cell type, which is this, SARS-CoV-2 infected lung epithelial cells, where we would like to predict what is the effect of this drug when you apply it. And that's very similar to when you have like neutral faces and smiley faces, and you use, for example, an autoencoder or a GAN to figure out what is the direction of adding a smile. And now, for example, in an autoencoder, you add this particular direction here to a neutral face, right? And you decode this new point from the latent space back to the image space, and out actually comes a smiley face. Okay, so that's exactly what you would want to do with drugs. So I know the effect of a drug on a particular cell type. I now want to figure out what is the effect of this drug on a new cell type. Well, let me do this in the latent space by just moving over the vector corresponding to drug effect over to the new cell type. Now, of course, all of this can only work if these vectors are aligned in the latent space, right? If drug A points into the drug A applied to cell type 1 points in the same direction as drug A is applied to cell type 2. Now, the amazing thing is that we can test this, right? Because we do have the CMAP data set, which has many, many drugs that are overlapping, that are applied to one cell type and also to the other cell type, so that we can actually look at how they are aligned in the latent space. Now, I can already tell you that in general, when you just use autoencoders, they're actually not going to be very well aligned. And they're basically just aligned as well as if you use, you know, just the original space itself. Okay, so now what we're going to use to make them better aligned is some insights that we just recently published in a PNAS paper on autoencoders and what kinds of functions autoencoders are learning. And so let me just give you a little bit of insights on that, because that's really the main insights we're using in order to build architectures for autoencoders that really align these drugs much better than the usual autoencoders. And so this is just very, very briefly what is this insight. So maybe a bit counterintuitively. So usually autoencoders, right, they go from a higher dimensional space, say images, to some low dimensional space here, back to the high dimensional space, because they're trying to get a low dimensional representation of your data that is informative, that captures as much as possible of the original data. Now we're doing something maybe not so intuitive. We're going to go from our high dimensional space, which could be an image or RNA-seq, to an even higher dimensional space, and then back to the original space. Okay, so we're embedding our data into a higher dimensional space than what you started off with. Now, you know, this is not usually done because it was believed, right, that this, I mean, it's obvious that this autoencoder could represent just the identity function. But it actually, what we showed in this paper is that it actually doesn't, okay? It doesn't, when you train it, it doesn't learn the identity function. It actually does learn some representation in a higher dimensional space that is actually an interesting representation, and in particular, interesting for aligning drug vectors. Now, why is it interesting? And what does it learn? Okay, so just for some intuition, what you can do is you can take an image. So the interesting thing about autoencoders is that you can iterate the map. I can take an image, out comes an image, I can put it back through the autoencoder and iterate it and see what comes out. Okay, so now the standard application of autoencoders, you know that if you take an image, you know, we took a training example, added a whole lot of noise to it, and you plug it through the autoencoder, out comes something that is denoised, right? That looks very similar to what you put in, but a little bit denoised as before. So it learns something that is very close to the identity function in general, but it actually doesn't learn the identity function. So if you iterate the map, you will see that out comes a training example, okay? So what this means is that autoencoders learn maps that are contractive at the training examples. Okay, so that whenever you put in something, it will actually output an image that is a little bit closer to one of your training examples. Now we have a proof for this, you know, in this setting here with being attractive to points. But now, of course, what is the intuition if you want to make vectors more aligned? Well, you know, these things, what we showed here is that it aligns new points to something that you've already seen before, right? By moving it a little bit more closely to a similar point, to a similar training example. Well, maybe this also works for vectors, okay? So maybe, you know, if you use an over-parameterized autoencoder, vectors that are already kind of similar will now become more similar to each other, meaning more aligned to each other. Well, and that's exactly what happens, okay? So let me show you here. So what we did here is we take two different cell types and that actually have the most drugs applied to them in the CMAP data set. And we look at each drug and we just measure the correlation between the effect of the drugs in the latent space, okay? So you would, if they're very well aligned, you would like to see it a lot of points at +1 or at -1, right? Then the vectors would be very well aligned. So what I'm showing you here is the correlations of these vectors in the original space, so without embedding into the latent space, and versus the standard autoencoder, this would be a lower dimensional embedding. And so what you see here is that, you know, all these correlations are along the diagonal, meaning that, you know, doesn't really matter. You can do it on the original space or in the latent space. Nothing really happened to these alignments. However, here in the second panel, if you actually use an over-parameterized autoencoder, you see that suddenly you have many more points up here and many more points down there. Well, what this means is that these vectors are actually much better aligned now. And in fact, the alignment is very similar to what you would get by just using the first and the second principal component. But that means you're throwing away basically all of the data, right? It has no signal in it anymore. You can basically not reconstruct anything that you had before. So these over-parameterized autoencoders, they actually align the drug effects, and they do this by not throwing away any of the data, right? You actually get perfect reconstruction in the end because it is over-parameterized. So that's quite nice. So now through this, of course, we can now identify the drug signature, right? So this, if we go back to this little picture, that allows us to now use all of the data together in order to identify the effect of a drug, which we will now correlate or anti-correlate with the effect of the disease, right? And so in that way, you can prioritize drugs. And what is quite interesting here, this is what I'm showing you here, is that you don't get just a random list of drugs, but you actually get basically two classes of drugs, these serine threonine kinases and these tyrosine receptor kinases. That I won't go into in more detail, but let me just show you. We also looked a little bit at the mechanisms of how these drugs actually work. So these drugs that were found actually have quite a few targets. So you really want to maybe look at, what are the targets that are maybe the most important, so that then we could also use drugs that were not in the original CMAP data set that are more specific to these particular targets that we actually identify. Okay, so you have a list of drugs, you have a list of targets. So how do we do this? Well, this is, again, a more standard approach, how people do this to prioritize drugs. And then I'll again tell you how we extend this. So the usual approach is you take your large protein-protein interaction network, right? We have already the genes that are differentially expressed by the disease and by aging. And now we just look at the subnetwork that has most of these genes in it. And we look at which drugs actually target genes that are central in this particular subnetwork that is the most affected by the disease. Okay, this is known as the interactome. And we're looking at the drugs that actually target nodes in this interactome. Now, I would argue that, this protein-protein interaction network is undirected, right? I would argue that just being central in such a network doesn't really mean anything. In particular, you care about the directions, right? I want to target a drug, a protein that has a lot of outgoing edges, in particular outgoing edges to genes that are differentially expressed by the disease, so that it can actually have an effect on these genes or these proteins. So you certainly don't care about drugs that look like this, right? That target a gene or a protein where there are a lot of edges that are directed inwards. You care about these kinds of proteins, right? Or these kinds of targets that have a lot of outgoing edges that point to or have downstream genes that are really affected by the disease. And so what we did is we added a causal analysis. So we actually took this undirected graph and learned a directed graph based in this case on single cell RNA-seq data. We did this in these A549 cells. We also did it in the cells that we actually have in our body, and these 82 cells, the normal epithelial cells. And in fact, in both analyses, you get the same target that is the most upstream to the most differentially expressed genes, which is this RIPK1. What is quite interesting is that actually if you remove aging from the analysis, okay, you don't filter out your genes based on aging, RIPK1 actually becomes completely peripheral. I should also tell you that if you remove the effect of aging, you actually get here a drug list that has all kinds of classes of drugs. So this is a really important filter for the drug list, and also, of course, here for figuring out the targets. And what is quite interesting, and this is nothing that we put into our analysis, there was this really nice paper that came out earlier this year where they identified which proteins in our body can actually bind to SARS-CoV-2 proteins, and RIPK1 was actually one of these. Okay. Now, RIPK1, we looked at it a little bit more in terms of the biology. It's a really exciting protein. I came to really like it, and we're currently looking at it a little bit more. And of course, this requires experimental validations now, which is things that we're right now doing. So with that, I'll end. We applied, or we developed this drug repurposing platform to look at SARS-CoV-2, but obviously this can be applied much more generally. I mean, all you need is this expression data of the normal state and the diseased state, and then all we're using are different data sets that are publicly available, like these protein-protein interaction networks, the CMAP data sets to get the drug signatures, and then the drug target data to figure out, what are the targets of each drug in this protein-protein interaction network. And so with that, let me end. And I'm just have here the references to these papers if you want to look at them. And thank you all for your attention.
Caroline, thank you. Thank you very much. I really didn't know that you've done so much since, I looked at the paper. It's very, very interesting work. And it's really lovely to see how, the new development of algorithms really make a difference here. And this is one of the points that we hope to show to the broader machine learning community. There is a huge space of method development which would have real impact and understanding of biological phenomena. And I think your work and the work of your group really is a beautiful illustration of this point. So we are kind of running out of time, but I would allow just one question if we have a question for Caroline. I've seen also that Bonnie very kindly replied to questions in the chat. Let me see if there is any. Okay, I will select one question. Are additional constraints required to make the over-parameterized autoencoders to land latent space where drug effects are aligned?
Yeah, so the interesting thing is no, right? So all we used is just over-parameterization. Now, of course, you could add, if this doesn't work and doesn't give you enough alignment, this is something that we're now playing around with. You could add additional constraints, but the interesting thing is that what we're also trying to prove is that this is sufficient, right? So this seems to work very well for the CMAP data set.
Thank you. So we have two other questions. If I may, I would just ask another one. So there are RIPK1 inhibitors in clinical trials. Are you looking to make and test these compounds?
Yes, so there are, and in particular, there are very specific inhibitors of RIPK1, which is why we really cared about identifying, some of the targets, right? So that we don't take these drugs that have so many other targets as well. And yes, this is something that we're trying out, but the experiments are a bit more difficult, right? Because you want to do co-culture systems that are actually of the aging lung and not just of the young lung. I think this is something that is really exciting going forward, and are experiments that people should be doing, and not just these standard co-culture systems where you apply a drug, because it seems like it matters, right, what the mechanical state is. So if you just take standard cells, they will probably not show the right effect.
So thank you very much, Caroline. If you can answer, there is some other questions that we unfortunately have time to ask. So thank you for an excellent talk.
Thank you.
Okay.